The Hardy-Weinberg equilibrium is a principle in population genetics that describes the relationship between the frequencies of alleles in a population and predicts the distribution of genotypes in the absence of evolutionary forces. The equilibrium is based off several different rules, but we mainly focus on the 5 types, which are large population sizes, random mating, no mutation, no migration, and no natural selection. If a population is in Hardy-Weinberg equilibrium, the allele and genotype frequencies will remain constant across the generations. Deviations from this equilibrium suggest the presence of evolutionary forces such as genetic drift, selection, mutation, migration, or non-random mating. The Hardy-Weinberg equilibrium provides a baseline for understanding and detecting changes in genetic composition with populations. For our experiment, we chose to observe the population size of our fruit flies. By selecting the amount of phenotypes and controlling the population size of each fly type to 5, Hardy-Weinberg equilibrium is violated because we are selecting the amount of flies that are going into a population. It's not random. And over a long period of time, evolution can occur due to this.
In this experiment, we will be using the species Drosophila melanogaster, which is also known as the fruit fly. Of this species, we will be using 2 phenotypes, the wild type or red-eyed fruit fly, which is the dominant allele, and the mutant white-eyed fruit fly, which is the recessive allele. To start, we will have 2 tubes which will provide for the populations. One tube will have 1 of each phenotype, and 1 tube will have 5 of each phenotype. Our hypothesis is that there will be more wild type or red-eyed fruit flies than there will be white-eyed flies at the end.